Hi everyone, my name is Brittany Lair. I'm a student with Grand Canyon University, and today I will be giving a presentation on phagocytes. At the end of this presentation, there are a couple of learning objectives that you guys should be able to do. You should be able to describe the different types of phagocytes and how they're made, explain the mechanisms by which phagocytes recognize pathogens, and explain the process of phagocytosis and how phagocytes destroy and break down pathogens. There are some key terms that I will be using throughout this presentation. We have granular and agranular leukocytes, neutrophils, macrophages, dendritic cells, phagosome, and opsonization. It really wouldn't be fair if we kept going on and talking about phagocytes if we didn't stop first and give some credit to the guy who discovered them. He was a Russian zoologist, so I will do my best not to butcher his name. It's Ilya Ilyich Mechnikov. He lived from 1845 to 1916, and he was best known for his work in immunology. He studied a lot with invertebrates, and it was actually in studying starfish larvae that he found phagocytes and phagocytosis. He would put thorns in the larva, and he would see that these things were kind of coming up and like quote unquote swallowing the thorns and digesting them. And it's this process that he was following. He coined it phagocytes and phagocytosis. He actually went on and earned a Nobel Prize for his work in cellular immunity, so pretty cool guy. All right, so what are phagocytes? Phagocytes, that is a blanket definition for a cell that can perform phagocytosis. So phagocytosis is a cell that can ingest and digest foreign particles, can be microbes, can be dead or dying cells, and break them down into their building blocks. So phagocytes are white blood cells or white blood cell derivatives. So we're going to be talking about the immune response. The left-hand side there is a picture of a macrophage. You can see like his little finger-like things coming out, and he's grabbing the bacteria and bringing it in. On the right-hand picture is a cartoon drawing of the same thing. So we're going to talk more about this, how the cells have these appendages that can attract these foreign particles, bring them into the cell, and they do their digestion within the cell. So as I mentioned, a phagocyte is a white blood cell, so it's going to have to do with the immune system. The goal of our immune system is to keep out foreign invaders. So if foreign invaders do happen to get in, then the goal of the immune system then is to kill them. So pathogens, we have viruses, bacteria, fungi. Our first line of defense against any pathogens, it's going to be our skin, mucous membranes, and then antimicrobial substances. As we all know, pathogens get past that first line of defense. We have a second and even a third line of defense. So our second line of defense is inflammation, fever, and phagocytes. That's our sweet spot for today. We're going to be talking about the second line of defense. On the right-hand side is just another depiction of phagocytosis. So you can see it has the receptors, the bacteria, and there's some digestion going on within the cell. We're going to go into all of that in some more detail. So phagocytes, being part of the white blood cells, are going to be constituents in our blood. So we have pluripotent stem cells in our bone marrow, and these stem cells become differentiated and more specialized and more specific in their function. So as that happens, we get our granular leukocytes and our agranular leukocytes. So the difference between our granular and our agranular, granular, when you're looking at them underneath the microscope after they've been stained, you can see the little granules. So like if you look at the eosinophil, that first one there, it kind of looks like a donut almost, like the icing, but you see the little dots everywhere. That are those are the granules within the cell that you're able to see underneath the microscope. Our agranular ones, you're not able to see the granules underneath the microscope even if they've been stained. So again, broken down into two types, granular and agranular. Our granular are going to be our eosinophils, our neutrophils, and our dendritic cells. And then our agranular are going to be our monocytes, which turn into our macrophages. So our different types of phagocytes, these are granular again, so like underneath the microscope you guys are going to be able to identify those grains, or those little granules, sorry. Our first one that I want to talk about is neutrophils. I want to talk about these first because they are the most prolific. So in our white blood cells, neutrophils make up between 60 and 70% of the white blood cell count. So its main function, phagocytosis, you can see on the left-hand side here, there's that picture. That blue that you can see is the nucleus, and then the rest of the cell with the granules in it. Our neutrophils, they are first to arrive to any infection. So they're highly phagocytic, and they can move. So they can exit the blood, and then they go into the skin, like into the tissue, they go through the epithelial cells, and they start to perform phagocytosis. So they can destroy microbes, foreign particles, and dead or dying cells. Mostly they consume bacteria. So their job is to mostly eat bacteria. They are made in great numbers, as you can tell, 60-70% of our white blood cells. And when they die, this is kind of cool, when they die, they actually become pus. So when you guys get like a cut and you can see it's like infected and it has like that sometimes clearish, whitish, yellowish stuff coming out, that's actually the neutrophils. After they did their job, they died. And macrophages actually come in and clean up after them. We'll talk more about that. But neutrophils, when you have an infection, neutrophils are the first ones there, and they're the most ones there. But at the end of the infection, neutrophils are actually very minimal in number, and there's far more macrophages. And I think that's the reason we just talked about, that the neutrophils die and they become pus, and then the macrophages now are left to clean up after the neutrophils once they die. The next type we have is another granular leukocyte, is eosinophils. So these guys are also phagocytic, but they're only somewhat phagocytic. These also have the ability to leave the blood, but their main function is to produce toxins that are deadly against a very specific type of parasites. So they're too small. The eosinophils are actually too small to ingest the whole parasite. What they do is they attach to the side and they release their toxins. It's peroxide ions that actually go into the parasite and kill the parasite. One cool thing to talk about with eosinophils, underneath the microscope, they have a really distinct look. So I think the picture on the right shows a little bit better, but you can see they have like this orangish, pinkish tint to their granules. It's really cool. You can see on the left-hand side too, they look a little bit more pink than the other ones. And they are in relatively small numbers. So they're only around 2 to 4% of our white blood cell count. So not as prolific as the neutrophils were. Dendritic cells. These are our last type of granular phagocytes. These are even less than the eosinophils. So the eosinophils were between 2 and 4%. The dendritic cells are 1 to 4%. So they're really rare. And I don't want you guys to confuse dendritic cells with dendrites. So dendrites are part of our nervous system. This is going to be part of our immune response. The dendritic cells are named so because they have those little appendages that they look like dendrites. But those appendages are what help them engulf the microbes and other invaders, foreign particles, things like that. And these guys are ideal antigen presenting cells. So on the outside of their cells, they present an antigen. This is actually important for their second function. They are phagocytic, so they can ingest and break down pathogens. But their second job is to alert the adaptive immune response. So these guys, with the antigen on the outside of them, they actually activate the B and the T cells, which are a different part of our immune response, which we're not going to talk about today, but they activate the adaptive immune response, which is different than the innate immune response, which is what we're talking about now. So they activate our T and our B cells. So that's their second function. The last type of phagocyte is our agranular, so it means under the microscope you can't see their granules, are monocytes. So these guys are bigger than the rest of the other ones. In comparison, they're quite large. And they still make up a very small percentage. 3 to 8% of our white blood cells are monocytes. So our monocytes do not become phagocytic until they leave the blood and enter the tissue. So when they leave the blood and they enter the tissue, they go from monocytes to macrophages. Macrophages are what perform phagocytosis. So there are stationary macrophages throughout the body, and then there's certain macrophages that are fixed to tissues, and then there are free-floating macrophages. So like the guys that are in the blood and then they leave and go to the tissue. So these are huge in comparison, and these guys are actually really cool. So they can eat up to 100 bacteria. So that's a lot. So these guys are big and they can do a big job. These are also the reason why we have inflammation in like the lymph nodes and things like that, because these guys are so big. They produce the inflammation in our tissues. All right, so phagocytosis. Now we're going to actually get into talking about what happens when a microbe, a foreign body, or a dead or dying cell needs to be ingested and broken down. So phagocytosis is actually super important not only for an immune response but also for a homeostasis standpoint, because all of these cells, even though they're dead, they still have these parts that can be used by the rest of the body. So it's really important for cell homeostasis to be able to break down those dead and dying cells and then use their parts again. Also the same thing with pathogens, being able to break them down, help the immune response. That's also another part of homeostasis, just keeping everything in check. So there are a couple of different steps of phagocytosis. The first step we have is chemotaxis and adherence. So chemotaxis is the attraction, the chemical attraction that is, it's a signal, right, that tells the cell where to go. So the cell is going to follow this gradient to either the site of an infection or to the site of the dead or dying cells that it needs to ingest. But chemotaxis is just, it's like a signal sent out, a chemical signal sent out to tell the cell, like, hey, over here, you need to come do some phagocytosis over here. So the cells will move up. How they're recognized on the microbes, this is the adherence part, or the attachment. It's facilitated by these pathogen-associated molecular patterns, which PAMPs. So you guys can see right here, PAMP. It's in the peptidoglycan of the cell wall of the microbe. And then in the cell wall of what looks like, I can't remember exactly what kind of phagocyte that is, but it's a phagocyte. There's toll-like receptors. So it's like that lock and key mechanism, right? So they attach. And once that adherence happens, that triggers the phagocytosis to start. And it also, not only does it that, it also recruits more phagocytes. So it sends out more of the signal, more of the chemotaxis, right? So more of the phagocytes are going to come to the area. So this one cell, as soon as he clamps in, he's going to start phagocytosis, and then he's also going to tell all of his friends, like, hey, we need you over here. There's infection or more cells that need to be eaten. So then all of his friends are going to come, and there's going to be a bigger reaction because of this. So opsonization, the phagocytosis will happen, but opsonization is a chemical, like it's a protein serum that coats the outside of the microbe, and it actually makes it easier for phagocytosis to happen. So it coats the microorganism in some kind of protein, and it makes it easier to ingest by the phagocyte. So that's opsonization. After adherence comes ingestion. So the pseudopods, which are just little fake legs, basically the wiggly parts around the outside of the cell, are going to extend and engulf the microorganism. They then meet and fuse. So you can see right here, it makes a phagosome. It binds with the lysosome, which has the package. It's the package that breaks down the microbe, and this forms a phagolysosome. Then this package is transported to the cellular membrane, and it dumps out the contents once they're digested into the cellular space. So that's the process of phagocytosis. The vesicles, the lysosomes, have enzymes that break down everything inside the phagolysosome, fuses to the membrane, and releases the package.